Hello, my name is David Henshalll and I'll be giving an overview of our workshop entitled Advances in Seizure Control by Targeting Novel Channels and Receptors. The idea behind this workshop was that we have a number of drugs that are effective at stopping seizures. We have anticonvulsants such as benzodiazepines, and we have a large number of anti-epileptic drugs. For many of these we know a lot about their mechanism of action. Unfortunately, so far we don't have any anti-epileptogenic treatments. And the purpose of the workshop was to present some of the latest research looking at very different targets for seizure control and anti-epileptogenesis. First presentation is by Professor Albert Becker on targeting an acquired calcium channelopathy. The work by this group identifies a T type of calcium channel which is increased after status epilepticus. Animals lacking this receptor are protected against hippocampal damage and develop milder seizures, and their work has now identified a transcription factor responsible for that change. It's called EGR1. It's an immediate early gene, and this offers a very new potential target. Second talk is by myself. This is work presented from our laboratory where we have been looking at the potential of targeting ATP-gated channels in the brain. These are ion channels which until now have not really been looked at in epilepsy. We show that drugs blocking the P2X7 class of receptor potently reduce seizures in an experimental model and can also be used to boost the effectiveness of lorazepam in refractory status. Next we have Marrowgar Coya, who presented his work on targeting neuropeptide Y receptors in epileptogenesis. Neuropeptide Y has mainly anticonvulsant effects, and using an elegant dual viral vector approach, he showed that neuropeptide Y delivery with overexpressing certain receptors could strongly reduce seizures in experimental models. And the last talk was by Professor Christophe Bernard on an NRSF-dependent HCN1 channelopathy. This work was based on the HCN1 channel, which is important in maintaining normal neuronal membrane potential. Downregulation of this channel is found in experimental epilepsy, and they look at the mechanism of this control and identified that the gene is in fact silenced by the neuron-restricted silencing factor, and blocking that gene reactivated that expression of HCN1 and reduced seizures. So in summary, the presentations in this session provide some really exciting new targets for seizure control. Some of these targets may also be potentially anti-epileptogenic. And the techniques used in these presentations encompass some of the latest tools we have for research, including epigenetics and gene therapy, which may be approaches to epilepsy treatment in the future.